right, folks, welcome to topic 2. So in the last topic, we talked about how we manipulate and analyze DNA. In this topic, we're going to talk all about how we clone DNA. And this is really important because this is how we actually identify the genes and what they do in the cell, or at least work on trying to identify them. So in this topic, we're going to talk about the principles of transformation, PCR, and DNA libraries. Here are your topic objectives. Make sure you have these clear in your head, and if you have any questions about these techniques, as I said, given the chance that you probably haven't done some of these, I expect them to be a little muddy for you, so let me know, and so we can talk about them a little bit more. So let's talk about the principles of transformation. Now, you may have done this in the lab before. If you've taken 201 recently, you've probably done it where you make E. coli glow. The cell lab, you guys should be doing this as well at some point. What the principles of transformation is that we take a DNA fragment, we put it into a recipient cell, almost always bacterial. It doesn't have to be, but we usually use E. coli simply because it grows fast, it replicates quickly, and it's pretty hard to kill. It works really well. It's an easy model to use. We insert this fragment of DNA into the cell, and the cell takes it up and then starts making the product of that gene. So that's the basic principles of transformation. So we're going to talk a little bit about how this all works in a little bit more detail, but the big key here is that by getting this DNA into the cell, we then have a mechanism to actually use that DNA for information, whereas in the laboratory we can't do much beyond analyze what the fragment actually is. So when we start with recombinant DNA, which is what we call the DNA that's been modified, we take DNA and we insert it into the genomic host of the DNA. So this is where that fragment would get incorporated into the genomic DNA of that E. coli. And what we do for this is we use restriction digests. We use those nucleases we talked about in the last topic, cut the gene and the host DNA, and then stick it back together so that they come together with this new sequence in there. Ligase then is used to help seal that up. So as you can see here, we have the two ways to do that. Now, big thing here is that the joining the blunt end with a staggered end or a sticky end is a little bit harder. So we usually try to do either the 2 sticky ends or 2 blunt ends. We don't generally like to do the staggered end because of the fact that it requires a little bit more work. But it all comes down to where are we trying to cut the DNA and what we want to insert. And that's really what the whole key comes down to, is if your sequence of DNA doesn't have the right fragments, you're going to be limited as to what nucleases you can use. We do the same thing with plasmids. So remember, plasmids are this naturally occurring circular DNA that bacteria make. And what we do is we'll open it up, stick in some DNA that we're looking for to be cloned, and then we'll close that plasmid back up. Now, the big thing to note about plasmids is that plasmids don't generally incorporate themselves into the host genomic information. They still use all the machinery, but they don't become part of the genomic DNA, which means it can be spit out from the cell. So it's a little bit different, but plasmids are a lot easier to work with, a lot easier to manipulate. And so we generally use plasmids a lot more in the lab, but that doesn't mean that we don't do recombinant DNA in the host. So one of the big things that we do with this all is PCR. And you know that PCR stands for polymerase chain reaction. And hopefully by this point in time, you've had a chance to do this in the cell lab. But if you haven't, but let's walk through how this works. So what we do is we start with our original sequence of DNA. And the whole point of this process is to make more DNA. So in step 1, we're going to heat the strands. And this takes the place of helicase and opens up the 2 strands of DNA. We're then going to add primers. We know we need primers so that we can start the polymerase action. So the primers get added. You have to have specific primers. So they have to be hybridized to a specific region in that sequence that we're looking to amplify. Then in step 3, we will elongate using DNA polymerase and a whole bunch of nucleotides and allow that to replicate. And then we just do the process over and over and over again. Usually, a lot of times in my laboratory experiments, we do at least 40 replicates. It just depends. And what this does at the end of the day is it takes a small amount of DNA and amplifies it so that you have plenty to work with for a variety of reasons, for a variety of applications. So now let's talk about DNA libraries. There are 2 main types of DNA libraries. There's the genomic DNA library and the cDNA libraries. C stands for complementary, and we're going to talk about that a little bit here in these next couple slides. So let's talk about genomic libraries. Genomic library is a very, very big process, very big tedious process. What we'll do is, and we've got some new technology that allows us to happen quicker, but the big key, but this is how we've done it in the past and how it's still sort of working together. What we do is we take the genomic sequence and we bust it up with restriction nucleases. Insert into plasmids, insert that into the E. coli or whatever host we're using, and wait and see what product's being made. Now, if we're super lucky, that gene that's inserted is going to make a product, and we now can say, okay, gene A makes product B. But that doesn't always happen because we know this now that not all the genes, first of all, to get lucky enough to cut the entire gene into the DNA or into the bacteria cell is very rare. And then the fact that we know that there are multiple genes that work on multiple products and with all the other aspects of it, we know that it's just not quite that simple. So that's why genomic libraries take so long. Another way that we can do this is we can take bacteria that contain the recombinant plasmids and then we can hybridize them to identify which plasmids have the plasmid of interest and then move forward with that. So that's how the genomic libraries work. It's a very long, tedious process. And as I said, we have some new techniques that are helping speed this up. But what it does is it requires us to break apart the entire genome and look at each part of it with different fractions at different times. The other thing that we have is cDNA. And as I said, cDNA is it's like the whole genome process, but it's not the same. What we do in cDNA is we take the mRNA product of the gene and we reverse engineer it to find the entire DNA sequence. Now, what do we know about this? This is problematic because we only use exons. We only use the part that we know will turn into the product that we're interested in. That means that the introns are left out, which means that we don't have a full picture of what this gene looks like. Now, that's not always a bad thing. It can be totally useful, especially if you just want to get your protein of interest and you want to make more of it. That's easy to do. But if you want to understand the full complexity of this gene, you're going to need the genomic library, not the cDNA library. So that's why it's important to understand. cDNA libraries are great because it gives you the down and dirty version, just a simple, this is what's going to create the product you want, versus the genomic, which tells you everything about it. It's like reading the Cliff Notes version of a book versus the whole book. So let's look at this again in the two different versions. So here you can see the DNA, the genomic DNA clones in the genomic library and the cDNA clones in the library and how this works. And we use reverse transcriptase for that cDNA building because we take that RNA, build it backwards into DNA. And so this just kind of shows you how the differences look versus exons versus introns, etc. As I said, you can use PCR for both processes, and it helps speed it up to an extent, but there's still a difference in it. And so the big thing to take home about genomic versus cDNA libraries is that they are not interchangeable. There are specific uses for one versus the other, especially if you're looking at protein products. The cDNA usually works pretty well. If you want more information about the entire gene and how it could be spliced differently, etc., that's when you're going to start talking about genomic libraries. So this is the end of topic 2. If you have any questions about this, please let me know. As I said, I expect these things to be a little muddy for you, so don't hesitate to let me know if you have any questions.